Hello, my name is Nicholas Cerrone, and I'm presenting the project, The DNA Damage Response in Whole Mouse Brains Exposed to 8th Grade Gamma Rays.
And I'm Matt Bramlow, and we worked on this project in collaboration with Julia Lentz, Erica Schaefer, and our faculty mentors, Dr. Wallace and Dr. Yasui.
In this project, a model mouse system was used to mimic radiation therapy exposure to the brain. Radiation exposure to the brain often has a side effect of altered cognition, known as brain fog. Female and male mouse brains were examined for persistent lesions indicated by the DNA damage biomarker gamma H2AX. The gamma H2AX biomarker was found to be persistent past typical DNA damage repair times, thus indicating potential chromatin alterations and reactive oxygen species activity. The aims of the project focused on the characterization and quantification of differences of gamma H2AX foci in male and female mouse brains. Extrapolation from the data can indicate a possible mechanism for radiation-induced brain fog. The experiment was set up using 5 male and 5 female BLBC mice that were whole body irradiated with 8 grays of cesium-137 gamma rays. 5 male and 5 female mice were assigned to sham control. After irradiation, mouse weight was monitored weekly. 15 days post-irradiation, mouse brains were sectioned and stained for gamma H2AX foci, as shown in the experimental design in Figure 1. The Zeiss LSM 900 microscope was used for super-resolution imaging of gamma H2AX foci.
We found that the consequences of radiation exposure did not include significant changes in the weight of the mice, except in the case of the male mice weighed 7 days after radiation exposure, where the weight of the irradiated mice was found to be lower than the weight of the control group mice, as shown in Figure 2. Additionally, no radiation-induced lethality occurred, and symptoms of adverse health effects like lethargy and reduced appetite were not observed. In Figure 3, mouse brain sections processed for immunohistochemistry and imaged using confocal microscopy can be seen. We have male and female brain tissue sections, both irradiated and unirradiated, from the hippocampus and the cortex. The green fluorescence foci indicate the presence of residual DNA lesions and therefore gamma H2AX, and the cell nuclei were stained blue, and images were acquired using a 20 times objective lens. It's also important to note here that DAPI staining was highly susceptible to photo bleaching, so nuclei staining was somewhat variable. And as Figure 3 shows, the gamma H2AX signal is much stronger in the samples exposed to the 8th grade gamma radiation dose, and increased number and size of gamma H2AX foci indicates a higher retention of DNA lesions in the irradiated mice, and it suggests a continued aggravation of the DNA damage response via inflammation response or reactive oxygen species generation. Additionally, we noted the presence of unusually large gamma H2AX foci in samples exposed to radiation, as can be seen in Figure 4b, but not in unirradiated samples shown in Figure 4a. And these images represent maximal intensity projections that allow for the visualization of the total number of gamma H2AX foci per cell. We also acquired AirScan super resolution images of gamma H2AX foci substructure seen in Figure 5, and these images may represent residual oxidative damage in chromatin. To summarize these results, the first trial of the experiment showed more gamma H2AX foci staining in both sexes in irradiated mice compared to control mice. In the second trial of the experiment, background levels were reduced by reducing the secondary antibody dilution, and this allowed for additional confirmation of increased numbers of gamma H2AX foci in irradiated mice brain sections compared to control sections. Future work for this project includes slicing more sections of brain tissue to test for microglial activation that indicates an inflammation response, which is a potential mechanism of DNA damage. This project constitutes an exciting collaboration between biology and psychology departments at NIU, which allow for the study of radiation-induced brain fog in a mouse model. We'd like to gratefully acknowledge the NIU SEF Student Engagement Fund for providing support for this project, as well as the National Science Foundation for helping with the acquisition of the Zeiss LSM confocal microscope.